everyone. So just before we get started, I just wanted to do a quick land acknowledgement. Because we are coming to you from the University of Alberta, we just wanted to acknowledge that the University of Alberta, its buildings, labs, and research stations are primarily located on the territory of the Cree, Blackfoot, Métis, Nakota, Dene, Iroquois, and Anishinaabe lands that are now known as part of Treaty 6, 7, and 8 and homeland of the Métis. The University of Alberta respects the sovereignty, lands, history, language, knowledge systems, and cultures of all First Nations, Métis, and Inuit nations. So our first speaker for today is Dr. Bradley Kerr. Dr. Bradley Kerr received his bachelor's in psychology from McGill University. He then went on to obtain a PhD in neuroscience from the University of London King's College in the UK, where he investigated the role of various immune factors in persistent pain. Dr. Kerr then went on to do postdoctoral work at the California Institute of Technology and at McGill University, where his work focused on studying inflammatory responses after nervous system injury. Dr. Kerr then joined the Department of Anesthesiology and Pain Medicine at the University of Alberta in 2007, and his lab now is aimed at better understanding the mechanisms of chronic pain after injury or disease. And without further ado, Dr. Kerr, please take it away.
Trying to unmute myself. Sorry about that. 5 years of Zoom and I still haven't mastered that. Okay, can you hear me? Okay. All right. Well, thank you, Arena. Thank you, Lana, for inviting us to do this today. So what I thought, well, what I was asked to do was kind of give a history of neuropathic pain research. And so what I put together is a very brief history of neuropathic pain research that is in no way exhaustive. And I apologize to all of the researchers out there who I've probably omitted or forgot to mention in some way over the long kind of arc and trajectory of neuropathic pain research. And what I kind of hope to do is really just to kind of walk through and tell you about the evolution of a lot of these models that we've used over the past 50 years to try to get a better understanding of the cellular mechanisms, the causes of these chronic neuropathic pain states. So the talk will be focused primarily on rat and mouse models, but to begin, I wanted to tell a little historical vignette. I don't really want to say that this is the beginning of pain research, but it's an interesting story and it's got a very kind of human, one of the very first kind of foundational studies looking at human chronic pain states. And so if I could figure out how to advance my slides, just give me a second. I'll introduce you to these two gentlemen. Excuse me. So this is Sir Henry Head, who was a neurologist working in England, kind of a late 19th century, turn of the 20th century. And he did some very pioneering work on nerve injuries and how these nerve injuries recover and the restoration of sensory function. And Henry Head was very interested in somatosensation very broadly. And the other gentleman on the slide here is William Rivers, who was a good friend of Henry Head's. And William Rivers really was the prototypical Renaissance man of the day, right? And you can see under his bullet points there that he not only was an anthropologist and an ethnologist, but he was a trained neurologist and actually then worked primarily as a psychiatrist. And he's probably most famous for his work during and shortly after the First World War, treating a lot of veterans in northern Scotland who were suffering from shell shock. And he was very famously, two of his most famous patients were these very famous poets who were very, very well known in England, Siegfried Sassoon and Wilfred Owen. And in fact, there's a Booker Prize winning novel called Regeneration, which was turned into a movie, which features William Rivers as one of the main characters. And Henry Head actually is kind of a background character in that novel. So if you ever want to read more about the life of William Rivers, that's a good source. But we'll get back to the pain research side of things. So Henry Head, as I said, was very interested in understanding somatosensory function. And in just before, in 1893, just before the turn of the century, they published a very seminal paper in Brain on the disturbance of sensation with a special reference to pain of visceral disease. And this, again, was a very foundational study of understanding the changes that occur in the nervous system when a peripheral nerve gets injured. But the one thing was, Henry Head was never really satisfied with the patient's reports. He actually quite often he was pretty blunt and said he didn't really believe his patients quite often in the things they were telling him. And so the thing he wanted to do was to kind of get his own understanding of what is going on when a peripheral nerve gets damaged. And so in the early 1900s, as you do, you have colleagues down the hall that are neurosurgeons. You walk down the hall and you ask them if they would mind sectioning your own peripheral nerve. And so that's what Henry Head did. In 1903, he had one of his colleagues, Dr. James Sharon, perform an operation where they severed two of the cutaneous nerves in his forearm. And then with William Rivers as his kind of laboratory assistant or his postdoc, if you will, they subsequently went on meticulously mapping the changes in sensory function that occurred and the regeneration of the nerve over the subsequent of the following 4 years. And so they documented, the painful sensations, the presence, the absence of stimuli, and really this is this foundational study where they mapped how long it took for sensation and normal sensation to return. And probably in terms of pain research, why this is really a foundational paper is in the paper, Henry Head makes this distinction between protopathic and epicritic sensory systems. And the protopathic is the one that dominates very early, and protopathic is a kind of a fancy term for very crude sensation. So this is really the stuff we think of when we think of pain, these kind of very broad, throbbing, aching, burning sort of sensations. And these really dominated in the beginning after the injury, and then eventually by the end of 4 years, these more fine, discriminatory tactile sensations, this epicritic type of sensory function was regained. And you can actually read this paper. You can find it online. It's very easy to access. It's on Brain. It came out in 1908. A Human Experiment in Nerve Division by Rivers and Head. Really a landmark paper. But of course, in terms of pain research, there are not a lot of people as brave or maybe as foolish as Dr. Henry Head who are going to, voluntarily have their nerves severed for such an experiment. So what the pain research field has really relied primarily on are animal models. And what we'll talk about mainly are the models in which rodents are used. So in the early 1900s, in most of the 20th century, the rat would have been the dominant sort of laboratory species of choice. This kind of changes in the 1990s when transgenic technology and we're able to begin manipulating genomes, and the mouse kind of takes over as kind of the primary model system to study pain. But I'll just highlight one kind of real landmark paper in terms of nerve injury research that was carried out by a kind of a seminal figure in pain research field, Pat Wall. And this was a paper from 1979 where he and Marshall DeVore kind of did this very broad characterization of all of these various peripheral nerve injury paradigms in the rat and documented the behavioral manifestations that occur. And they coined this term. What they noticed is that depending on the severity of the injury and when the injuries were so severe as in which the nerve was completely separated from its target tissue, that the animals actually had this tendency to begin attacking their own paw as if they didn't know it was part of their own body. So there was this tendency for the animal to attack its anesthetic limb. And they coined this term autotomy for this phenomenon. And this paper is a kind of a meticulous kind of description of the time course, how long it takes for these behaviors to come on, and then the degree, the amount of this behavior that occurs, depending on, again, the severity of the injury. And the paper is important in a sense because what it sort of told us is that they were able to do very careful careful electrophysiological assessments in these animals and understand the different changes in the firing properties of specific populations of sensory neurons. And they determined that the emergence of this autotomy-like behavior is really triggered by this abnormal barrage of activity from these primary sensory neurons. And the correlate to the human kind of pain condition is this phenomenon known as anesthesia dolorosa, which is painful numbness. So you can't actually feel your hand or your lip, but you're sensing this kind of ongoing burning sensation. And a lot of people have also kind of uh, made parallels to the phenomenon of phantom limb pain with some of these models. So, most of these early studies were doing these full, complete transections of peripheral nerves, like the sciatic nerve innervating the hind paw. But, aside from the ethical considerations, and there's not many vets in today's universities that would kind of allow these kind of studies to go on anymore. And then the more practical question is you're not really able to ask these animals or present these animals with any stimuli and see any responses because the limbs are basically deafferented. They're cut off from their target tissue. And so one of the members of this team highlighted here is that Seltzer spent a number of years trying to develop a rat model in which you could then cause an injury in the peripheral nerve, but then actually ask the rat about various types of stimuli. You could present the rat with a heat stimulus and see how the response changes, or you could present the rat with a mechanical stimulus and see how their responses changed. And so this is the development of what we know as the partial sciatic nerve injury model. It was developed late 80s. The publication probably was 1990 officially. And we all kind of knew it as the Seltzer model. And this is the one that was very prominent in the late 80s and 90s because what it did was instead of taking the sciatic nerve, which, sorry, there's my cursor, you can see right here, instead of kind of sectioning it completely and cutting off the hind limb from the spinal cord and the DRGs up here, what Seltzer and his group said was, well, if we only do a partial injury, we cause some damage, and then we still allow this animal to have some intact fibers that progress and make their way into the spinal cord up to the brain, and you can get these long-term behavioral changes. And it's a very simple surgery. It just involves exposing this big sciatic nerve in a rat. Now we've translated this into mice. And you just put a simple ligature and you tie off approximately half of the nerve to generate this state. And down here, this is taken from their abstract. You can see, within a few hours of the operation, the animals have very prolonged for several months guarding behaviors of the injured paw, and they lick often, which they take as a sign of potential spontaneous pain, which is kind of a valid sort of modeling of the human neuropathic condition. And importantly, in this injury model, none of these rats actually show this self-attack, this autotomy behavior. So that was very good in that sense. Now, one of the disadvantages of this model, though, is the variability you're going to have. So yes, it's a very easy surgery, and you can perform lots of them kind of in an experiment or a day. But there is this disadvantage of the variability because, again, from rat to rat, the size of the nerve, are you taking exactly the same half, the same amount? And so you can have variable injuries, and then it becomes a little difficult from a scientific perspective to kind of control that every subject in your study is experiencing the same level of injury. And so to answer that question of variability, other groups went on and developed a model that became very kind of commonly used in pain research. And this is known as the spinal nerve ligation model. And it was developed out of Texas in the lab of Jinmo Chung. And so again, these models all kind of take the name from the person who invented them. And so we often refer to this as the Chung model of neuropathic pain, the spinal nerve ligation. Instead of taking the sciatic nerve and causing the injury down here in the main kind of trunk of the peripheral nerve, what Jinmo Chung and his group did was they moved very close to the sensory neurons themselves in the dorsal root ganglion. And what they did was they took one of the spinal nerves, the L5 in this case. I think they have variations in the paper where they try all three or just one. And so the way the model has evolved, and today it's mainly just one of these spinal nerves. And so you take a suture again and you ligate, you tie off this spinal nerve and you section it. And the advantage you have in this setting is that now you are certain that you have caused an injury to every axon that innervates the L5 dermatome. And the other advantage is that your other DRGs, like your L4 and L6, you know those fibers are all uninjured. And what you can start asking questions about is in the nerve itself, where you have uninjured L4 fibers mixing together side by side with injured L5 afferents, you can ask, how does that change the way the uninjured afferents are going to respond to subsequent stimulation? And so that's presented down here in their abstract again. The model has these two unique features. The procedure is stereotyped, and again, that's talking about the consistency of the injuries you're going to get from subject to subject. But then the levels of injured and intact spinal segments are completely separated, and you can allow for independent experimental manipulations of those. So you can ask questions about changes that occur in uninjured afferents, et cetera, et cetera. Now, this is a much more difficult surgical procedure than the Seltzer model. And so it didn't necessarily catch on as well. It does have its advantages, but it does require a very practiced microsurgeon to be able to access this particular spinal nerve and perform this very tight ligature and section this. So it's not as easy to perform. And so it makes kind of implementing large scale experiments very, very difficult. And so the answer to that difficult SNL surgery was to develop a model where you could get a very consistent, very clean type of injury model that's very repeatable from subject to subject, but is much less, requires much less microsurgical expertise, if you will. And so this was developed back in the early 2000s, and this came out of Clifford Wolf's lab with his postdoc at the time, Isabel Dekerstedt. And they developed what I would say has now become probably the prominent, most commonly used peripheral nerve injury model to understand neuropathic pain, and that's known as the spared nerve injury. And so now instead of being up here in the spinal nerve or down at the main trunk of the sciatic nerve, what Isabel Dickerstedt and Clifford Wolf did was they moved down, further down the sciatic nerve to the point where the nerve begins to branch into its various specific arms. So we could see the sural nerve here, the tibial branch, and the common peroneal nerve here. And what they did then was, again, very similar to kind of the Chung idea, where you're going to try to have a nerve injury where you have a mix of injured and non-injured. They're going to tie off and ligate two of those branches, two of those three branches. So the tibial typically and the common peroneal, get the ligature, get fully sectioned, so that you know now that every tibial axon has been injured, but all of your sural nerve fibers in this sciatic nerve are intact. And then that leaves some ability for behavioral responses and sensation. You don't really get that autotomy behavior that we saw with the full complete transections, but you get a very prolonged and very pronounced, very repeatable type of nerve injury that then generates this very chronic change in somatosensation. And that's showing you down here, taken again from their abstract. It results in a very early change in sensory function. Within the first day, you can see animals, their responses to various stimuli are altered. And again, mechanical, thermal, all the various modalities that you would want to test and understand are showing changes. And so these are the kind of the primary traumatic nerve injury models. So these are very good at understanding what happens when nerves get directly injured and directly kind of damaged in some physical way. But again, in many clinical conditions, the nerve is never fully transected. There's very rarely or it's much more rare that to see a complete transection of an axon in a human peripheral nerve. More often than not, nerves are compressed, or there's some maybe a tumor pressing down on a bone, which then is triggering some inflammation. And so to model that, in response to those kind of questions, Gary Bennett in his lab developed an alternative to these frank kind of transection models. And this is known as the chronic constriction injury model. And this was actually developed kind of around the time of the Seltzer model itself back in 1988. So instead of actually going in with a ligature and completely tying off the nerve and then using sharp scissors to cut axons directly, what Bennett and his team developed was this model in which you basically just constrict the nerve itself. So you take four sutures, and they're generally of a kind of a catgut material, which then triggers this inflammatory reaction. And the idea is to tie these sutures around the nerve just to the point at which the nerve gets a little bit of constriction. It gets just pinched ever so slightly. That doesn't frankly or overtly cut the axons, but triggers an injury state and then generates a long-term inflammatory reaction around the nerve. And again, this model has this advantage of maybe much more closely resembling a lot more of the human pathologies you see in a lot of these people with peripheral nerve injuries or nerve injury diseases. And the post-operative behavior of the animals is very characterized and very characteristic. They indicate all the same kind of sensory changes that those other models have. They show hyper-responsiveness to noxious stimuli, but also hyper-responsiveness to non-painful stimuli, so this allodynia phenomena, and also evidence potentially of the spontaneous pain. And the one thing that we should point out is that these injuries, this injury is, again, because the axons aren't completely severed and prevented from regenerating, there is a certain amount of regrowth and regeneration, and these animals will eventually recover back to kind of a normal threshold after a period of time. So that being said, taken all together, all of these models do kind of what we want them to do if we want to try to understand human neuropathic pain, because the feature of human neuropathic pain is its chronicity. It's not a pain that, you present a stimulus, there's a change, and then an hour later it's back to normal. These are states where the sensory changes are lasting weeks to months to even up to years. And all of these models do induce these very profound, very prolonged changes in responses to mechanical stimuli, to heat stimuli over here. And these are just some examples from the spared nerve injury model. As far out in that original paper, they tested up to close to a year, and the animals were still showing hyper responsiveness compared to simple crush injuries where you can see the responses kind of regain back, heading back up to more normal processing. Now, again, these are all sort of mechanical injuries to peripheral nerves, but neuropathic pain arises in a whole host of diseases and disorders. And it turns out we have models that can mimic many of these different conditions as well. So, for instance, a lot of patients undergoing chemotherapy develop peripheral neuropathies in response to those chemotherapeutic drugs. Well, people have developed animal models to study that type of pain state that's generated in response to chemotherapeutics. And those models involve just taking a rat or a mouse and giving them the exact same drugs you would give a human cancer patient, and giving them those treatments, and they can generate, as we can see over here in this graph, very prolonged, profound changes in sensory responses. Again, a chemotherapeutic model is a model in which the pain will be restored after a period of time. Not only chemotherapy, but diabetes. There's various ways of studying diabetic-induced neuropathy. There are pharmacological ways. You can give rats or mice, you can alter their diet. You can give them drugs that kill off pancreatic or islet cells to induce a diabetic state. And then you can see that their sensory function is altered. And there's also genetic lines of mice as well. HIV is often associated with a neuropathy and a chronic pain state. And so people have found that you can take elements of the HIV virus, just one of the coat proteins, with this protein called GP120. And if you just put it around a peripheral nerve, you soak a nerve in this HIV protein, you can trigger changes again in pain sensation that are very prolonged and very profound that again model the state that you're trying to study and try to understand. So those are in the peripheral side, but we also have models for central neuropathic pain as well. So various stroke models can also trigger prolonged changes in mechanical sensation and heat sensitivity. Spinal cord injury models, which are shown here in these graphs, these are have been developed in which you contusion injuries in the spinal cord. And many of those, the primary outcome of that research is really to understand injury responses and regeneration of the spinal cord. But one of the consequences of spinal cord injury is prolonged neuropathic pain. And it turns out these models actually do kind of give the same kind of outcomes where you can see prolonged, profound sensory changes in these mice. And then I'll just give a little shout out to our own sort of focus of our research. We study chronic pain in the setting of multiple sclerosis, and we have animal models in which we can mimic the disease process that's very similar and looks a lot like MS. And we're finding that, this animal model of MS triggers very similar sensory changes that you see in clinical populations. So let me just finish here now. I'll wrap up. What I've been talking about are these models that have this advantage in which we can present stimuli to the animals, and then they tell us how their sensory thresholds are changing over time, and whether potentially we can use them again to test therapies to see if we can make pain better. But one of the key features of neuropathic pain and human neuropathic pain, if you kind of go back to the Henry Head sort of analogy, is that the pain is always there. It's not always in response to a poke or a pinch or an application of a heat source. It's always there, and there's always this spontaneous or persistent pain state. And these tests and these assays are not maybe getting at that sort of side of the neuropathic pain condition. But we do have ways in which we can sort of study ongoing or spontaneous pain, and it really capitalizes on facial expressions. So this image here, I think all of us would probably assume he's being pinched or something painful is happening to him. We have a very distinct grimace or facial features when we are experiencing something painful, whether this is evoked or even spontaneous. Now, these features kind of can be translated into a rodent's face, it turns out. And so Jeff Mogul was the real pioneer of this work, developing what is known as the grimace scale. He developed this first in rats, but now as on this diagram here, we could see it's been applied into mice. And again, what it's doing is taking some of these very characteristic features of a painful response, the pinching of the eyes, the bulging of the cheeks, and ascribing them to rodents. And so in these experiments, instead of applying a stimulus, poking it with a von Frey hip filament or presenting it with a heat stimulus, you just have to watch the animals very carefully. And usually you do this with high-speed video cameras, and then you go through the footage and you kind of score frame by frame, and you're looking for orbital tightening, for instance, or the flattening of the animal's ears as an indicator of their state that they're in and whether there's a prolonged or ongoing pain state. Now, this is very labor intensive, and where it's kind of evolved into the 21st century is now obviously with artificial intelligence and neural networks. And so this is where I'll end. I'll just tell you about Mark Zilka's lab and how they've developed a platform in which you can take those mice or rats, you can film, get your high-speed video camera, and instead of having to sit and have an experimenter evaluate the facial expressions of the rodent, you can feed the images into this neural network that they've called PainFace, and then the PainFace will evaluate these features for you, essentially. And so here's just a little description. This is from one of our pilot studies, again, using that MS model, the EAE model, and where you can see this program is measuring features about the position of the ears, whether the eye is open, the whisker orientation. And you can see an animal over here that has this disease is starting to show elevated features of this grimace, if you will. And this is just some very early data, some preliminary data. We're continuing to do this work, but we can see that this mouse model of MS is inducing what we would determine to be a pain state that is kind of always there. It's we don't really need to poke it or ask it how hot they think this feels. And so now we can again use these models as platforms to see, can we make this pain go away with our various treatment and drugs. So that's a whirlwind tour of the animal models we use in pain research. I'll just summarize here, and then I'll hand over to Bruce, who will lead us into the human neuropathic pain state. But basically, I think I want you all to kind of take home today that we do have at our disposal, we have a lot of means to study neuropathic pain, and we can model the symptoms and outcomes quite well, I think, in a laboratory setting. I do want to put a cautionary note. Henry Head is kind of a hero of mine, but maybe again, somewhat foolishly. So please, self-experimentation is very ill-advised. There's very few ethics boards who would allow Henry Head to do the experiments he was doing in this day and age, and I wouldn't really advise it for anyone. But again, the chosen approach, really, it depends on what you want to model, what disease you're interested in, what are the injury factors that you want to study, and then what's available to you in your laboratory setting. Do you have expert microsurgeons who can carry out the SNL model versus the SNI model, for instance? And really this question of traumatic injury versus non-traumatic injury, that will also help determine which model you want to apply to your research. But again, well, probably the take home today is that I think all of these models do a pretty good job in reliably generating these long-term chronic changes in sensory function that we then now, as I said, can use as our platform to begin trying to either understand the pathological causes of these changes, but also as a platform to test novel therapeutics. And finally, the last little point here is that we have ways of going beyond just simple stimulus response questions, but we can also now employ methods like the grimace scale or pain face to assess these bigger sort of pain features, such as chronic or ongoing pain. So I'll end it there. I'll hand over to Lana, I believe, who will hand over to Bruce. And thank you for your attention. And are we taking questions now, or should we do that generally at the end?
I think we'll save questions for the very end. So I guess I'll just introduce Dr. Dick and then we can take it away. So yeah, the next speaker is Dr. Bruce Dick, who is a clinical psychologist by training and also a professor at the University of Alberta. His doctoral research focused on the electrophysiological responses to tonic and chronic pain at Dalhousie University, and his postdoctoral training took place in the Department of Pediatrics at the University of Calgary and Alberta Children's Hospital. Bruce provides clinical services to youth at the Pediatric Chronic Pain Clinic at the Stollery Children's Hospital in Alberta, and is the Chief of Psychology in the Multidisciplinary Pain Center at the University of Alberta Hospital. He also provides clinical services there to adults. His research interests include a number of studies associated with pain research and management across the lifespan. And with that, I'm going to give the floor to Dr. Dick.
Thanks, Lana, and thanks so much, Brad, for your great presentation today. So I've been asked, recognizing that there would be some people here today with lived experience of chronic pain and neuropathic pain, to speak to the effects on the person, both of some of the physical effects, but perhaps primarily on a person's life experience, including their mental health as a psychologist. I don't have anything to disclose today, but I do like to throw this in at the beginning. We're grateful to be here, and it's exciting to watch this pain research network kick off and go. Brad kindly provided his slides to me a while back, and I finished my talk over the weekend, but I added this slide this morning. And just thinking about the day and thinking about the chance to do this with Brad, I want to acknowledge Barry Finnegan, who is our past chair, who hired both Brad and me some years ago. And I don't know that I've ever told you the story, Brad, but I sat with Barry a few weeks before his retirement, and he was just reflecting on his career. And he said one of the things he was proudest of was bringing the basic science group into our lab, specifically naming Brad, because Brad was the first hire in that group. We have Stefan Bork and other great people now too, but Barry said one of the best things he ever did was hiring you, Brad. So I threw his slide in this morning about half an hour ago because I think he'd be really happy. His goal was to bring together basic science and clinical science, and it took a little while. But Brad is leading a CIHR grant, looking at he didn't talk about it today, looking at neuropathic pain in amputees using both an animal model and a human model with Jason Plemmel and some other great people. And I think Barry would be happy to know. I hope he's having a good time in Ireland or wherever he is now. But I think Barry would be happy to know that this is happening today. And I noticed Glenn Baker's here today. Hi, Glenn. Glenn was a mentor, agreed to be my mentor when I was first hired a long time ago. I'm glad to be here in Edmonton with Brad and others, and I'm really grateful for the opportunity to be here today. So I've been asked to talk about the psychological impact of neuropathic pain and the impact of neuropathic pain on quality of life. So I'll do that fairly broadly. Some of the data that I talk about looks at chronic pain more broadly than specific to neuropathic pain, but I will talk about mental health and how chronic pain impacts mental health. Many of you are experts as people with lived experience with more expertise than me. With that, I'm going to talk a bit about the health related and social costs to chronic pain in Canada because I think this impacts all of us, most importantly, people who live with pain. But as a society, chronic pain is a big deal. And with an election coming up, maybe there's some food for thought here if you want to interact with your MPs and MLAs as time goes on. So a few of the key points that I want to share today is that chronic pain is complicated. Neuropathic pain is complicated at every level of this model that we call the biopsychosocial model. It is fundamentally a medical issue for people who live with pain. And it also has big impacts on people psychologically and socially. And we're social creatures. And so that's huge. It's important for the individual who lives with pain, but it's important for all of us as members of a society in which chronic pain has a major impact on our health system. It can be a challenge to treat and manage, and most importantly, a challenge to live with for people who live with it. And I want to provide just a very brief discussion at the end about the fact that there are effective ways to manage it. Brad's talked about the development of treatments, of medical treatments and pharmaceuticals. Every time we who work with people in clinics hear about the development of these new pharmacological options, we're very excited and we're very hopeful for the future because of good work from Brad and people who do what Brad does. Now, this is a very broad slide. I won't spend a ton of time on it, but I appreciate this because it just shows the impact of pain globally across the world, anywhere that you go. The most common reason why people seek medical care is pain, and this happens across the world. Literally billions of people are affected by headaches, for example, and it's consistently been one of the leading causes of disability across the world. So it's big. A little closer to home in Canada, this comes from the Chronic Pain Task Force, published a few years ago, essentially is suggesting that 1 in 5 Canadians lives with chronic pain. 2/3 of those Canadians report that their pain is moderate to severe. And this is big as we have an aging population. These numbers are only going to rise. As well, since the pandemic, there's growing evidence that these numbers are actually increasing post-pandemic for a variety of reasons that we won't necessarily get into today, but maybe we can talk about later during questions. One last thing about the cost of pain to society. It's very expensive. For those of us also who don't experience chronic pain, we want to think about this as we think about how health is managed, because if we live long enough, chronic pain is coming for all of us. All right. And so if you look across the world, about 1 in 5 people experience chronic pain. About 30 to 40% of these people have neuropathic pain specific to what we're talking about today, Brad and me. This has a pretty big impact in terms of costs across the world. And what's important in Canada, and I'm going to show you some data from Europe in just a second here, People with pain access health services 3 times more often than matched controls. This is really big from a cost standpoint. It's important for all of us to think about this as we think about resource allocation. So if we look at, this is a really helpful publication. It's a bit dated, but really important. Again, this is that 1 in 5 number in Europe. But the unfortunate part in what was a really well um, described study, less than 2% of people with chronic pain ever see a specialist, at least in Europe. And I think that's pretty common here as well. Um, the important part for all of us is that when chronic pain is present, all aspects of health tend to be worse as well. It predicts worse health physically and mentally. Okay. And again, as I mentioned earlier, the cost of this is absolutely massive in Europe, as is the case in the rest of the world. So then, despite this, it's unfortunate that it's difficult to treat, and there are good reasons why it's difficult to treat. We won't really get into this today. Brad and colleagues and others across the world are doing amazing work, but it's difficult. It's difficult to develop these things for a variety of different reasons. Finally, from a data standpoint, in terms of the impact on society, the impact in terms of disability, this is Canadian data from 2012. If you look at it as we age, the numbers associated with disability rise tremendously. I mentioned earlier in the bio that I submitted that I work with young people with pain and folks later in life. This is important to all of us because this will touch our lives or the lives of people around us in significant ways. And this level of disability rises over the lifespan. This is very, very expensive as well. As we think about neuropathic pain and what it does for the individual who lives with pain, it's complicated. It has massive impacts across various different measures in life for the person, for those of us trying to help these people live their best lives and have the best quality of life possible. The question for all of us will be, where do we start? And I'll talk a bit about the implications of this as we go, but we feel sometimes that this is where we go. So we pick a thread and we go. Now, for all of us, as we think about pain, there are different kinds of pain. Today, specifically, we're talking about neuropathic pain, but I'm going to talk about these three ends. To most of us, nociceptive pain or acute pain is what touches our lives on a daily basis. Happens all the time. Almost everyone experiences nociceptive or acute pain. For the purposes of what Brad and I talk about today, we're focusing on neuropathic pain, pain related to nerve injuries or nerve diseases. I'm going to talk a little bit about nociplastic pain, which is Brad mentioned in a couple of his slides, central pain and changes in the central nervous system. We deal a lot with that in the folks we're fortunate to work with in clinic. But for today, I'm going to try to focus as much as I can, as Brad has, on neuropathic pain. Now, Brad referred to neuropathy, and I think these are slides that I borrowed from Brad years ago that we use in some of our patient sessions, but this is normal tissue. Scientists have found a way to selectively stain nerve fibers that are involved in our experience of pain. This is in healthy tissue, and this is tissue from someone who experiences peripheral neuropathy that Brad mentioned in his talk previously. So you can see that I'm not a basic scientist, I'm not an expert, but even to my untrained eye, the difference between these nerves, where if you even touch this tissue with a feather, feels like a feather. For many people, you touch this tissue with a feather and it feels like you're raking broken glass over their skin and that their skin is on fire. So for those of you who live with pain, people with lived experience, this is a big deal. The tissue itself is relatively healthy. The bones and connectors there are healthy, but the experience of pain is massive and can be debilitating for people who live with pain. Now, our international association that provides us with definitions, expert-based definitions, neuropathic pain is defined as pain that's caused by a lesion or a disease of the somatosensory nervous system. It can relate to peripheral nerves, central nerves. Brad gave us an excellent description of this already. In terms of the lived experience, I use the word perceptual, but I should probably just have put the word lived experience for those of you who live with chronic pain. It's a severe problem. It has an impact on our emotional responses, essentially the areas of our brain that are involved in our emotional experience are highly connected with the parts of our brain, our pain system, our pain networks. That affects our mental health. And unfortunately, to date, for many people, these problems are intractable, which basically means it's difficult to manage. That's a part of the broader IASP definition. And so as we work in clinics to try to help people, and as Brad and his colleagues do the work that he does, all of the work that we all do is aimed at improving this, at reducing these impacts on people's lives. Now, these are the origins. Brad spoke to some of them. I take this from a 2020 publication. Um, these are many of the ways the people that we see in clinics come with nerve traumas, compressions, can be associated with vascular diseases, neurological diseases, infections, metabolic problems. It can be related to drugs. Brad talked about chemotherapy models before, hereditary issues, all kinds of things, right? And these treatments related to all of these things, while there are broad-based treatments for them, for us, we treat the individual specifically to their circumstances and their medical needs as well. So what do we see? Like this is where the neuropathy comes from, but what do people with lived experience? Brad talked about these things. So there can be localized pain, widespread pains that spread, that can be referred pain. It's often perceived as burning pain, electrical pain, shooting pain. You can have paresthesias that refers to sort of numbness and unusual experiences perceptually. Brad talked about allodynia. So basically this idea that you take a feather and you rub it across that skin and you experience pain when you wouldn't expect that. So the pain threshold has been changed. He mentioned hyperalgesia. So having a painful stimulus that becomes amplified for the sufferer because of these nerve changes. And you see all kinds of central changes, hyperreactions of the nerve system. And these are relevant to the individual sufferer because people come to clinic and their primary care providers in the primary care networks take an x-ray, do a nerve test, have a look at the tissue and say, tissue looks fine, your x-ray is normal. And the person in the chair says, Doc, I'm on fire. And my sleep is terrible, my mood is off, my quality of life is awful, and everything looks pretty good. So there are all kinds of things that happen. Okay. These are really, really important. These are the scientific terms for the awful things that people go through. Now, for us, what happens? What do we see? Well, we see people presenting with pain that is far in excess to what we would expect, given what you see in typical medical tests. If you look at the test that Brad does, Brad's work and the work of other basic scientists show us what's happening at the level of the individual nerve, in the spinal cord, and in our brains. That pain leads to changes in mood. Commonly we see higher levels of depression and anxiety. So depression increases. Work by Gord Asmundson and others talks about depression increasing fivefold, so 500% increases in people who live with chronic pain. Anxiety will increase eightfold, so you see these 800% increases. And so it's easy sometimes for people to think that these folks just have mood problems. Their sleep is disturbed. Sleep is the universal health provider. A colleague and I gave a lecture this morning about sleep to people who live with chronic pain. Sleep is absolutely massive. If we deprive people of sleep, we affect every aspect of health in their lives. And chronic pain, neuropathic pain, is a primary disruptor of sleep for people who live with it. Of course, this impacts physical function. You've seen the disability data that I've shared with you earlier today. Neurocognitive impairment is something that's key to my research program and the research program of many of my colleagues. We're very interested in how pain disrupts the brain. We've been looking at this for decades. I'm getting old now, in terms of how pain impacts our attention, our working memory, storage into long-term memory, and broad-based cognitive function issues. And quality of life is a very broad measure. But interestingly, a colleague of mine, Dr. Seifi Rashik here, has done some great work with our group and others, looking at the fact that quality of life in people who live with chronic pain is often lower when measured than people who experience cancer, HIV and AIDS, Parkinson's disease, catastrophic diseases that affect people's lives in massive ways. This doesn't mean that those patient populations don't have significant issues with reduced quality of life. But living with chronic pain and invisible disease seems to bring something that's kind of special in a bad way for these people. Now, I have another good colleague, Mark Simmons, who's been our medical director in our pediatric chronic pain program for years, who sometimes describes the impact of pain and chronic pain, neuropathic pain in people's lives, a bit like a bike wheel. So he went to United Cycle here in Edmonton some years ago and asked just for a donation and an old beat up bike wheel. They gave him one and he took it home to his garage and took a sledgehammer to it and beat the living daylights out of it, saying that it's a good model for what pain can do to a human life. And sometimes this neuropathic pain starts to take a central role in terms of impact and what it does. And we have the bike wheel in our clinic. If you're ever in the hospital, come track me down. I'll show you this great bike wheel. And we've had kids in the program talk about the impact of their pain on their lives, going to school, spending time with friends, their sleep, their health, their ability to take part in physical activities, to dance and do fun things. We look at it oftentimes in this way. Pain interrupts all kinds of things in life, our energy level, what we can do, our social contacts, remembering that our social connectedness is one of, if not the best predictor of health. It affects our mood, our stress level, and at a basic science standpoint, the activation of stress is massive in a person's life who lives with pain, can lead to increased anxiety, as I mentioned before, and poor sleep. And while we're still, experts like Brad and others are working on pharmacological methods for managing pain, what the role that we take or the approach that we take broadly, our physiotherapy colleagues and my psychology colleagues and I, because the effects between these factors are bidirectional, we work with people to help them improve their sleep. And we offer sleep management sessions and a sleep course. We work on managing mood, pacing, and increasing physical activity, which is great for health as well. Movement is medicine. And then reducing the activation of the HPA axis and what that does, the stress response system, the impact of stress hormones is massive as well. That's an entire other talk for Brad to give on another day. So what do? We take this picture and we focus on 5 key areas, and I'll show you the details of this as well. So we work with people to reduce physiological arousal. Now, this is about behavior or behavioral medicine, but it also has an impact on how our brains react, on how cells function. Simply put, we teach relaxation exercises. We teach meditation like mindfulness practice in people's lives. We do a lot of work with activity regulation. I would argue that the key player on a chronic pain team is actually the physiotherapist and helping people to move because movement is medicine. And so our colleagues in physio are massively important in what we as a team do. Mood management is key because the brain plays such a crucial, essential role, and pain has an impact on mood. And thereafter, there are all kinds of great studies that show that the intensification of depression and anxiety creates a problematic role for the sufferer, reducing quality of life, but also activating the brain in unhelpful, unhealthy ways implicitly. I talked about the HPA axis and bringing down this response. Reducing physiological arousal is a part of that. But increased levels of cortisol cause increased firing in pain fibers, and thereby stress affects people on a day-to-day basis in clinic. And then again, I'm interested in attention and how our brains function, but mindfulness practice and mindfulness work can be crucially important in helping people to manage their pain. So here's the broad picture. What does this look like in the clinic? Well, oftentimes you'll see in the clinical literature, in the clinical world, we talk about the 3 Ps: physical therapy, pharmacotherapy, and psychological therapy. And I would put them in this order. Not that any of them are unimportant, but helping people to gently, safely move in a way that is healthy for them is massive. Here's my 5-minute warning. Pharmacological therapy is crucially important, but it's difficult to develop drugs. It's expensive and it takes a long time. That's always a work in progress. And then I'll talk just briefly here in the next minute or so, and then we'll go to questions about psychological therapy and what we do clinically as psychologists. So 3 Ps. Interestingly, we teach a lot of behavioral medicine around moving, managing sleep, reducing stress, improving mental health, and living our best lives. One of the mantras of our program is life despite pain. How do you live your best life despite the fact you have this lousy neuropathic pain that has come from changes in our cells, changes in our systems, changes in our brains? Nutrition is obviously a key part of that as well. Our education sessions that we run, we look at these things in 3 different or 4 different phases. We do pain neuroscience education. We talk about healthy coping strategies. This is largely based on cognitive behavior therapy. In our intro programming, we talk about managing flares and setbacks, and then maintaining this on a day-to-day basis in people's lives. And it looks like this. Okay, so it looks teaching what's happening, building skills, having a plan, and changing how we live our lives, and making this a consistent, productive approach to living your best life when you have this lousy, invisible disease. Calvin and Hobbes, the text for whatever reason hasn't shown up here. This is my favorite cartoon, but Calvin has this great gem where he says, sometimes I find that it's easier to live my life when I reduce other people's expectations. I think a big part of what we do is helping people work in society, teaching society about pain, and teaching individuals maybe also to change some of their expectations as well. That you can live a full life, but sometimes it's about living our lives in a different way. Finally, I'll talk a bit about some of the other courses that we run. We have a drop-in class for alumni from that initial CBT group. We've developed a 10-week mindfulness program, a 12-week dialectical behavior program, positive psychology. This is actually mislabeled here. The PERMA model is actually from Seligman's model here. A 10-week program on positive psychology, a mindful self-compassion course taught by Mark Simmons, my colleague, a CBTI group for helping sleep. It's an 8-week course that we ran this morning. And broadly, we integrate all of these things along with individual psychotherapy to help people again live their best lives. These are offered by Zoom and in person as well. And here's a painting. We're fortunate to work with Alberta Education with the provincial government to help young people get credit, high school credit for taking our pain courses. And this is one of the final projects from one of the kids who took the course, integrating some of the skills. And I think it's just a really great image and a great symbol of what people with pain can live with, and what can be done that life can be better. And I'll stop there and happy to answer questions with Brad.